Hello, everybody, and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Coaster, and we're happy you're here today. We will be talking about myosin modulators for the treatment of cardiovascular diseases with Brett Morgan today. Welcome to the show, Brett. We're glad to have you.
Thank you, Dennis. My pleasure to be here.
Yeah, we already had like an amazing pre-discussion. Really enjoyed learning so much from Brad already in the pre-discussion. Now, without further ado, I am going to introduce our guest, Brad. So Bradley Morgan, he's currently serves as the Senior Vice President of Research and Non-Clinical Development at Cytokinetics in South San Francisco. He and his team are inventing and developing medicines to improve the health span of people living with cardiovascular and neuromuscular diseases of impaired muscle function. And earlier in his career, Brad held several roles of increasing responsibility at Pfizer, where he was the lead inventor of several new chemical entities that entered clinical trials for the potential treatment of diabetes and obesity. And at Cytokinetics, Brett is a key inventor of numerous potential medicines with novel mechanisms of action to treat diseases such as heart failure, hypertrophic cardiomyopathies, and diseases of impaired muscle function. We will hear about two of those treatments more today. Brett has published over 50 peer-reviewed scientific articles and book chapters, and is the inventor on over 95 issued US patents. Brett obtained his PhD in bioorganic chemistry from the University of Berkeley in California, University of California at Berkeley, I should say, and did a postdoc at Caltech. So yeah, thank you, Brett, for being here today. Very impressive CV and very impressive career you had so far. So yeah, we're really happy to have you today.
Thank you, Dennis. It's actually an honor to be here today.
Awesome. Yeah, and in the good old Flash Talk tradition, we want to start with some warm-up questions. So Brad, can you talk a little bit about what inspired you to start a career in drug discovery?
Yeah, I'd say there were two major things. And first, I'll give a shout out to my undergraduate university, Illinois State University. Go Redbirds. Because I entered there as a biology phys ed major, thinking my dream job was to be an athletic trainer for the Los Angeles Dodgers. But in there, my organic chemistry professor, Bernard Reiter, was super inspirational and really was the first class that I ever had in my life that made me think, right? And it really inspired me in sciences. Not knowing what I wanted to do with that, so I ended up switching to chemistry and getting a degree there. Not knowing what I really wanted to do, probably the second thing was getting a job at Eli Lilly straight after undergraduate, and seeing that how a science nerd that really loved the science could impact multiple people's lives in drug discovery was really inspired me to really get further training both at Berkeley and at Caltech on there. From the get-go, knowing that I quite likely wanted to go back into drug discovery from that initial work at Eli Lilly.
Yeah, no, that makes total sense. The teachers and schools we attend, mentors we have are really so important. Yeah, and we just talked in the pre-discussion about how you were throwing 2,000 pitches to younger kids. So I'm not surprised about that Dodgers trade. Yeah, so my next question would be, and again, I might already take a guess at your answer. What is your favorite molecule and why?
So you'd already know, Dennis, the answer to that one. And that is one of the molecules I'll be talking about today, Aficamten. And one of the reasons for that is I used to always tell folks in my group that enjoy the day you nominate a clinical candidate, because that's the best the molecule's ever going to look, right? Because once you get a molecule into development, all you find out is what's wrong with it after that. And it goes down. I can say Aficamten has been the exception to that rule, because everything that we designed into it for it to do, how it to look, has seemed to recapitulate through development into the clinic. And we haven't really learned anything that we didn't know. So that's why it's my favorite molecule, because it's broke the cardinal rule that I used to always tell people.
That's great. And yeah, we definitely look forward to hear a little bit more about the discovery story today. that I think is also super, super exciting. Yeah, with that, what are you most excited in drug discovery today?
You know, what really thrills me today is, because I started, in drug discovery in 1983, right? And if you go back from then till you look today, the amount of tools that are available to the base, the amount of knowledge that we know is so much greater. And the tools keep building on it. And the way I believe these tools will help us move faster, but more importantly, I think they'll help us move with better quality through the system. So hopefully we can get less attrition to that as we move through the system. Now, I don't think any of these tools are a panacea. I'm a big fan of AI. I don't think it's a panacea, but I think it can help us do things more efficiently and more effectively, and maybe even think of different questions we need to ask as we're moving forward in that. And so just the new tools that are available, utilizing these tools, trying to understand them, trying to figure out where they fit into drug discovery, I think. And if folks that are younger, if they're not making use of all these new tools, you should, because you're going to be obsolete in 5 to 10 years.
Wow, yeah. Well, I didn't even know you started your career in the pre-Lipinski area. But yeah, it's and Lilly. I mean, Lilly in the 80s and now like two completely different companies, right? Like it's definitely fantastic to see. Yeah, great. No, I think the new tools, and I definitely second you on that. Is we're in a lucky generation today to have all these tools at our disposal, so to say. Um, if you could go back in your career and give your younger self some career advice, what would you say to yourself, Brad?
I would say celebrate those small successes, because those of us that have been in the industry know how hard it is. I mean, we get into this because we want to make a difference to patients, but you have to get to the market to make a difference. And that's a long haul. And if you don't celebrate those little successes along the way, it's going to be a long career, because some people will never get to the market. And that's okay, because you're building for others to build on to have those successes. So celebrate the small successes along the way.
That's really phenomenal advice. Yeah, I can only definitely second that. It's like otherwise you're going to be very depressed. Like there's a
Yeah,
lot of failure in our industry, but we're learning from this. It's like iterations. It's the next iterations. We're learning
absolutely.
from those things. So
And it's all building on one another.
exactly, absolutely. Yeah. With that, last but not least, what do you like about Drug Hunter?
So I'm going to go back to graduate school for this. And so you got to think, I was in graduate school in the mid 80s. So this was before, electronic looking for references online was not real commonplace yet. Um, so I remember, like, I'd spend Sundays was my library day in graduate school. And you're always looking for things that can improve your program. And uh, one of the things that I used to go was you go to the library, but you'd find an article you weren't looking for. And it'd be kind of cool and interesting. And you'd end up going back and doing things. And ended up on these like wild goose things where I learned a ton. I ended up spending like 8 hours in the library and never get to the problem I was solving, but I ended up with learning a lot about a lot of different things I didn't intend to. Drug Hunter is a way for me to do that kind of thing almost automatically now. And I'll go to your GLP-1 like expose and what you had on that as an example. When I kind of popped up, I'm like, oh yeah, I remember we were talking about small molecules for GLP-1 in the 90s in my Pfizer days, and had no clue of how to do that. And you've got this very concise article on, and it shows the evolution over decades of how you get there from, the peptide point of view, and how that's now parlayed into small molecules on there, and still how hard it is. I just saw a former colleague, Dave Griffiths, give a talk on Pfizer's, small molecule for GLP-1 that they pulled, out of development now. And that's been taking decades in the making to get there. And it reminds us all of how hard this business is. And we got to be in it for the long haul.
Awesome. Yeah, thank you so much. Yeah, Brett, I will, before we get started, just 1 minute, show people how the page looks like. And you're bringing up a fantastic point of serendipitous discovery, right? So we actually have those 3 case studies here. And if you hit the refresh button, you find 3 new case studies. So that basically gets to the point that you were just talking about, Brett, serendipitous inspiration. And then you can click into those studies here as well. We have the orforglipron story. You mentioned danuglipron being pulled. We also had positive phase 3 data for orforglipron, so we updated our case study here. And if you wanted to use Drug Hunter a little bit more deliberately, you can also go to the full text search here and type things like, clinical updates in a different month, for example. So clinical updates in December, I wanted to show. Every month, we're doing FDA approvals, new clinical trial initiations, basically when the FDA accepts NDAs. And here, that is obviously what I wanted to show. Aficamten's NDA was accepted in December 2024 with a PDUFA date set for September 25. We'll hear more about that. Clinical trial initiations, you will find on there positive as well as negative readouts of clinical trials. And really, we go more in the science. As you see the molecule structures here. You see a little bit more background on the target, why this is important, what is the background. So you basically see all these things here on Drug Hunter on a monthly basis. And with that, I will hand it off now to Brett, and I'm very excited to learn more about Afi and Omi. let's call them by the abbreviations before I misspell or mispronounce the name. And yeah, I invite Brett to share his slides and basically teach us 1 or 2 lessons here on cardiovascular diseases.
All right. So I want to get started first by acknowledging the hundreds and hundreds of people and over 2 decades of work that went into these 2 molecules. I unfortunately don't have an acknowledgement slide with everyone on it because I would miss someone. And rather than missing anyone on this and all the I get the pleasure of presenting this work to you, but this work is done by my hundreds of colleagues at Cyto that have touched this over the years, that have really put blood, sweat, and tears into this to make it all happen. So at Cyto, we have really focused on the sarcomere. And the sarcomere from Z line to Z line is composed basically of 2 major elements. That is the thick filament and the thin filament. The thick filament is basically myosin, polymeric myosin, which is the engine or the motor that causes muscle contraction. It takes chemical energy in ATP and converts it to a mechanical force that drives every muscle contraction. The thin filament is kind of the transmission or the actin that's decorated with the tropomyosin that kind of blocks the actin, the myosin binding sites, and the troponin complex, TC and I, that regulates the tropomyosin. This is all regulated under nerve control. The nerves actually stimulate that bring calcium into the system. Calcium binds to troponin C, causes troponin T and I to actually open the tropomyosin and allow binding sites for myosin and for it to progress along that. As the calcium leaves and those white flakes you see leave, then those binding sites close up and you get relaxation. This happens with every movement of the muscle, happens with every heartbeat that we do. All of these proteins, you can find small molecules that regulate, bind to these, and actually modulate various sections of contractility. In that, for cardiovascular disease, there are many diseases that really are associated with both a reduction or an increase in contractility of cardiac myosin. For example, those associated with low contractility, diseases such as heart failure with reduced ejection fraction, what you see from a typical normal heart is you see kind of a dilated heart, a thinning walls, atrium that has been expanded like a balloon from a back pressure filling from the heart on there. On the flip side of that, you can see diseases that are associated with a overcontractility of the heart. Key among those is hypertrophic cardiac myopathies. There are many genetic conditions to hypertrophic cardiac myopathy. These genetic conditions can cause, at the myocyte level, hypercontractility of the heart. That hypercontractility over time causes a thickening of the walls, causing a lessening now of the ventricles. So you have a lot of back pressure build up, and you have less, again, all of these stimulate a heart failure. You have less blood that's being able to be pumped out of the heart because of these conditions. So we went in initially decades ago with the hypothesis, if you can normalize contractility, you can treat the underlying causes of many of these diseases where contractility may be driving the disease. Oop, wrong way. So to get down to the root problem of cardiovascular disease and heart failure, this is a major epidemic in the country. Now, we have a lot of good ways, much better ways to treat heart failure than we have had in the past, especially with SGLT2 inhibitors and now GLP-1 coming online to treat this. Much better we have, but there's still a niche for other things. We still need a lot more to treat this. It's a high burden, it's a high cost. Most importantly, when you look at people diagnosed with heart failure, 50% will die within 5 years. Now, this is a worse prognosis nowadays than most cancers. You know, the leading cause of death is cardiovascular disease in the country. There is still a huge medical need to fit this, both for not just longevity, but for a quality of life with these patients. So decades ago, we really had a system of looking at how do we find molecules to functionally screen the sarcomere. And we were able to really, what I would call the first company to really industrialize the process of screening for compounds that modulated sarcomeric function. And our initial screens were what we call recapitulating this sarcomere into a test tube, where you take the actin, a component, the S1 fragment of myosin, the troponins and the tropomyosin, recapitulated in a test tube. And now what you're looking for is the ATPase. Knowing that hydrolysis of ATP by myosin is directly converted to force, and that should be proportional to both force. And in the 384 well plate, you can now find molecules that both activate or inhibit, this ATPase to bring that along. This is robust. This was developed here. It's used now by even other companies to search for functions in the sarcomere. And you can do this not just with reconstituted sarcomere, but with things that are myofibrils or small three-dimensional pieces of the sarcomere in itself to do this. And we've screened, now probably even closer to 20 million through these systems. This is a number that's 17 million is probably a few years old here. But more importantly to that is it's not just having not having systems you can find hits, but having a system that you can recapitulate this function as you get to more and more complex systems that can mimic the human disease and show that what you're doing at that basic level that can recapitulate into the whole functional system. And we've built these systems over the years. As I've mentioned, the reconstituted sarcomere and myofibrils, very flexible biochemical assays for different functions along the way, both for MOA and for screening and for SAR in that, but showing that ATPase recapitulates into our force measurements, both in native muscle fibers, either skinned or whole fibers in various systems, and more recently in engineered muscle tissue that you can now engineer, and also in human-like muscle tissue that can be even disease modified in this way, and into functional organs, either whole organs like Langendorf hearts, or into animal models and animal models of disease. Going from a high throughput, to low throughput, but the coherence of biology is faithfully reproduced throughout all these systems. And you can go one to the other to then actually get into humans on that. One of the nice things about the sarcomere when you talk about species is these individual components are highly conserved across species. So we've seen very rare instances to where things we've seen in a bovine system or a porcine system do not look the same in a human system. So it's the exception rather than the rule for it not to cross species. So very different from other targets that people may be familiar with. So going back to then now this whole system for disease, let's start with heart failure with reduced ejection fraction and low contractility as our first story today. So going into this, we knew that small molecules can increase heart function and can increase contractility. And this was well known in the literature that by regulating really pKa phosphorylation indirectly can cause increases in calcium transients. And those increases in calcium transients causes an increase in contractility. So from indirect means, from dobutamine, which, beta agonist, or milrinone, a PDE3 inhibitor, you can increase these calcium transient cycles and increase contractility in the heart. Unfortunately, that comes with its consequences because it's really a high energy demand. You're also increasing heart rate, you're decreasing directly blood pressure, you're increasing the oxygen demand of the heart and decreasing the efficiency of it. It's also known that by increasing heart rate through increasing calcium transports, you can increase arrhythmias, and these arrhythmias can cause an increase in sudden death. So at the end of the day, you can make people feel better, but you can you actually decrease their survival because of the propensity for arrhythmias and sudden death. So we thought that if you could go downstream of calcium directly at the sarcomeric level to increase contractility, could we then do that in the absence of all these other changes and increase perhaps the efficiency of the heart, or at least not decrease its efficiency and not have these arrhythmias and have an effective drug for heart failure with reduced ejection fraction? So starting from screening, we ran, that I've talked about, we found this, basically this diaryl amide, nitroamide, not really too attractive from a drug-like perspective, but from a biology perspective was a super attractive lead structure in that it had really low micromolar potency and low micromolar in cells in terms of being able to dramatically increase fractional shortening of 135% in cells. And we believe that was really relevant because we just probably want to tweak it a few percent into humans for a disease. So we never want to get to that high. So we're even talking about relevant concentrations to treat human diseases with this. It was also very selective from the cardiac sarcomere right from the get-go, as opposed to fast skeletal, smooth muscle, or control. Now one has to remember that slow skeletal myosin is exactly the same as cardiac myosin. So you're never going to get selectivity over slow skeletal on there. But for fast, it was completely selective. It also seemed to be on target because in whole cells, we could show that we could increase this contractility very rapidly, and we saw no perturbation in the calcium transients in cells, no effects on action potential, durability on that. But obviously, extraordinarily poor physical properties and not very drug-like looking. So to make a long story short on the optimization, on a couple years worth of effort here, I'll walk through kind of how we modified this to get to Omecamtiv Mecarbil. And first, it appeared from our initial SAR that we needed a electron withdrawing group, at the 12 o'clock position. We were able to switch out the nitro for a fluoro, maintain activity. The other thing that we were able to do was to switch out the amide for urea. We wanted both an IV and an oral agent at the beginning of this. And so we wanted something that was very stable in solution over long periods of time for an IV, but yet also could have this half-life for an oral drug as well. And the amide really improved the hydrolytic stability over long periods. And it also gave us a much more, I would say, permissive SAR on the right side and allowed us to do more with the molecule as well. The second thing was really to try to get activity in vivo. And probably we didn't have enough free concentration in vivo to really see the effects of our labors at this stage. And we saw that basically by de-aromatizing the left side, you could maintain potency. And now you got the free concentrations higher, that we saw really our first in vivo activity in 534, and first signs of really that we could have at least an IV drug. We still had high clearance for an oral drug, but this could be first signs that this could be our first IV drug that we could use to enhance contractility in drugs. One of the issues we found with this upon further study is it also at super therapeutic concentrations appeared to have a direct effect on blood pressure and lowering blood pressure. Well, there's two ways you could increase contractility of the heart, right? You can do it directly at the heart, or you can decrease the afterload, right? And that will increase contractility. And so by lowering blood pressure. So we didn't want to do, there's plenty of drugs that do that now, all the neurohormonal agents that do that. We didn't want that to be kind of a side effect of the drug, and we wanted something that was strictly on target. So we found out that really SAR on the west side of the molecule, we could remove. We found out through really complex pharmacology that our off-target effect was due to KHP channel activity. We were able to remove that and remove any off-target effects of this blood pressure lowering by all the SAR very quickly on the SAR on the left side, west side of the molecule. Matter of fact, the opposite stereochemistry of 534 removed that activity, but still had some on-target activity there. And came up with really our next, what I would say, IV molecule for increasing contractility in 296. But it still was far from an oral drug in that its clearance was quite high, albeit it had very good, up to 20–30% oral bioavailability. So we thought if we could just solve the intrinsic clearance issues in microsomes, that we could probably come up with a good oral and IV agent. And the biggest revelation came was really in switching out this west side completely for now a drug that gave us enhanced solubility. By actually blocking this nitrogen, we could remove any CYP inhibition liabilities that we had as well, and dramatically improve the pharmacokinetic properties and have really our first IV to oral indication drug in 138 on this. And if you'll notice, we've gotten rid of the electron withdrawing substituent that we thought was important to the SAR. Turns out that as we improved the physical properties and got rid of that what we thought from the early SAR was not really true from this. So always go back to your assumptions. However, to kind of fill in that SAR, we went back in and filled in kind of just our initial electron withdrawing hypothesis, and found that really putting a fluorine in the 6 o'clock position turned out to improve both the PK and the potency of our compound, and gave us then Omecamtiv Mecarbil, which was our compound we ended up going into our human studies and through phase 3 with, and currently in phase 3 trials for this. And to just really show you in a nutshell of how these changes improved kind of the in vivo, I'll start with 296, which is really our first compound to work both in rats and dogs and in models of heart failure as well. And here we're looking at increased fractional shortening of the heart versus just as our time component. And this is just be from a constant infusion IV on this. And you can see that to get to really statistically significant increases, which is around the 120 mark, that it wasn't until we got to concentrations of about 4.5 micromolar and doses about 4.5 mg per kg per hour, you can see robust increases in fractional shortening, probably much more than is actually needed to have a therapeutic benefit to that. If you go now to 138, you can see both our doses, the concentration somewhat lowered to get to those concentrations. And now as we both improve the potency and the PK, You can see we've lowered both the dose and the concentrations needed to get to those robust increases in rodents for contractility. How did this improvements in contractility then do in phase 2 in patients of this? And I'm a chemist, so a picture is always worth 1,000 words to me to actually show the meaningful benefit, the potential meaningful benefit to patients. And on the left, you can see this is a patient with stable heart failure with reduced ejection fraction. And this is a cross section through the atrial, left atrial and ventricle in this patient. You can see the left atrium is expanded. The atrium is almost like a balloon, and when there's a lot of back pressure, that balloon overfills and can expand. You can also see in the left ventricle, you can see how thin, how kind of it's a little bit dilated. You can see how thin the walls are. And you can see even this wall is not really even contracting in. It's almost like that pressure is pushing it out because that contraction is gone. And finally, the mitral valve, because of these pressure abnormalities in this patient with heart failure, you can see the flutter. And definitely you're not getting that flow of blood through that heart. After 24 hours of drug infusion with Omecamtiv, one can see now, although far from normal, this left atrium is now much reduced, suggesting that these back pressures have now gone down. You can see the walls are thicker in the ventricle on there. You can now see this ventricle wall actually contracting more normally. And more importantly, this mitral valve actually looks a lot more normal in its function. So you can see from the hemodynamics a real improvement in this patient that's with stable heart failure. That was then enough to go to phase 3, where we ran our phase 3 trial with Omecamtiv, which was a success in that we had improvement of primary composite endpoint in first-time heart failure and heart failure death relative to placebo. Now that improvement was relatively modest, especially those who are looking at the drugs like SGLT2 that have recently been done, but yet was statistically significant and clinically meaningful to patients. Equally as important that the adverse event profile, was balanced between treatment arms. So we saw this improvement in patients and the absence of any of the safety signals seen with these things that improved through the calcium transients. So our initial hypothesis was correct. We can improve patients' lives without the detrimental effects on that. So that was seen. Unfortunately, a p-value of 0.025 was not enough for a single trial to get approval, and we need to run a confirmatory trial for approval of that. But what was pretty exciting from this data, now this is an 8,000 patient people data, is when you looked at the treatment effect versus the ejection fraction. And as you get lower and lower in the direction of ejection fraction, the treatment effect between placebo got greater and greater. This is different from any other drug that's out there to treat heart failure today, including the newer agents, as far as we know. That a drug that actually worked better on patients more in need, where most of these drugs actually either don't do any better or start to fail in these patients. So with that information in hand, we decided to really advance this to a pragmatic phase 3 trial for confirmation of activity. We believe there is still, for that patient population, a high unmet need. It's larger, it's growing, and despite newer things like SGLT2s, these patients remain at significant risk. And probably 18% of maybe the 7 million plus patients with HFrEF are in this category with the electric ejection fraction of less than 30%. So we're in the midst of running COMET-HF right now to really look at the severely reduced ejection fraction patients. So we think this drug will be a real lifesaver for patients in this category of drug and really meet an unmet need. Now, to really turn the story now to the other side of the coin. So it's kind of how we first got into the heart failure arena with augmenting, but there's additional ways that you can think about, well, what about the other side of the coin and people with hypertrophic cardiac myopathy or certain subsections of even HFpEF populations that get to their disease through the root cause of that could be an increase in contractility. And so can we kind of dampen that down with this? And the answer to that is, well, yes, it's actually been done now in this day and age. And there's a first-in-class cardiac sarcomere inhibitor in Mavacamten that was done. Now, this actually came about from a program that was initiated at Cytokinetics conceptually in 2007, when the idea came about to, why don't we look for inhibitors? In 2009, we ran the screen that found the forebearers of Mavacamten. We had we ended up doing other things with our resource and not working in that program. Had a couple people on our SAB come to us and say they wanted to launch a new company, wanted to do it within the walls of Cytokinetics, a company that later became called Myocardia. That was launched in that first year of life within the walls of Cytokinetics. And during that first year, a compound was discovered that was later called Mavacamten. And that has since reached the market, is now marketed by BMS, and it's demonstrated very profound benefits to patients with OHCM. So it's really awesome that this happened. But it does have some issues with these patients as well. And some of those issues are some of the properties that it has. And it's got a very long half-life in patients of 9 days. It also preclinically, showed signs that it could have DDI issues. But probably the long half-life and its steep concentration effect curve makes it really difficult to titrate in the right dose to have those benefits on there. So we went in really thinking that, well, great mechanism for disease, can really help these patients. Can we find a better molecule to do this at the end of the day? So we went in really with 3 primary objectives. One of those were to decrease the half-life, but yet have a half-life long enough for QD dosing, but yet reach steady state within 2 weeks. So probably we were shooting from between 1 to about 3 days on that half-life in order to do that, and to have really minimal peak to trough in there. We wanted preclinically no substantial CYP induction or inhibition of that to improve some of those preclinical parameters. And also we wanted to try to have a larger therapeutic index. And how do you do that preclinically? We thought really looking at kind of echocardiograph across species and look at kind of having a more shallow concentration effect curve could perhaps giving a larger pharmacodynamic window may translate into a larger therapeutic index. So those were kind of our objectives that we moved into the program with, to try to find a next in class and what I like to think could be a best in class for this target. So we started with an initial screening hit that was a singleton out of the library on there. Not too unattractive molecule. It probably, when you're talking about potencies, we already knew that you don't have to be too potent to modulate these initially. On this ligand affinity, not quite where you want to be, but relatively in a drug-like zone on that, but definitely microsome food on there. So we wanted to go in, and the beauty of this is we saw, we really were looking for things that were selective for cardiac, especially over smooth muscle. We didn't want to have any smooth muscle activity inhibition that could have blood pressure effects or things, and that was it. The other thing that was good about this is we could see, and again, in rat ventricular cardiomyocytes, we could see at high concentrations of robust, way too much inhibition of that in the absence of any effects of calcium transient. So again, on-target mechanism. The other thing that we found out about this very early on, about this class of molecules, is they bound to a different site than Mavacamten. Mavacamten was not, there's a pan-myosin inhibitor called blebistatin, which was first discovered actually as a smooth muscle myosin inhibitor, reported as a smooth muscle myosin inhibitor. It turns out that it hits a lot of type 2 myosins with probably equal affinity on that. And there's a crystal structure of it in a fungal myosin too. And we found out that although we had selectivity for smooth muscle, we were actually competitive with the blebistatin binding site. on that, where mavacamten was not. So we knew kind of right off that we had a different mode of binding on there. But we also knew that oh, well, great, there's an X-ray crystal structure of blebby, at least in that. We can kind of start modeling in and try to think of where can we find extra, extra ways to get both maybe affinity, but more like specificity for our molecules. And found that really by reframing this nitrogen and moving it out to the linker, we could maybe pick up this hydrogen bond, the same hydrogen bond that blebistatin picks up with the analogous leucine backbone chain. We might be able to pick up with aficamten. Lo and behold, the compound was made. It led to a 3x improvement in potency. And now you had this amide linker that was easy to kind of switch and mix and match that made the chemistry much easier in terms of the SAR. When we removed any potential analyte moieties in terms of that may be metabolites from this as well. The first thing, then it came actually quite quickly, the fine tuning from here. That was the big revolution. The fine tuning came in learning that first of the R configuration had all the activity of this, where the S, quite fortuitous. That R configuration also improved the PK parameters more than the S. In that was serendipity and a great discovery to have in this, but was not planned, I will have to admit, on this. And at the end of the day, looking back at the R1 modifications, it turns out that the small R1 was the best in tolerance, and the whittled down to those 271, 274, and of those aficamten having the best properties at the end of the day and advanced to the human clinical trials. So, as I've said, we knew that this was different from what was out there in the clinic, is that it was competitive with the blebistatin site, in that you can see aficamten can compete out blebistatin and this normalized fluorescence where mavacamten could not. And again, different site can this have different pharmacology at the end of the day. So since then, a lot of X-ray crystallography has been done to these, and we've shown that both for omecamtiv and mavacamten actually have the same binding site to myosin, kind of at the hinge region. It's kind of interesting. One's an activator in phase 3, one's an inhibitor on the market for HCM, but they have the same binding site, but probably stabilize slightly different conformations of this lever arm that may I would point you to kind of these communications. Everything I'm talking about today is public, by the way, published. I'm just putting it together in a story on that. Where aficamten binds to what we call the blebistatin site, which is kind of farther out in the myosin binding region on that. And as I mentioned, that blebistatin is not selective for smooth. So what's going on? Well, through our collaboration with the Ann Hadoos laboratory, we were able through this actually was painstaking and took probably a couple years to get this X-ray structure, but we were able to really confirm that yes, it indeed is binding to this same pocket as blebistatin. Indeed, it is actually forming this hydrogen bond, right? This NH hydrogen bond to the leucine in the backbone of this. But more importantly, we can find explanations as to why we now have this selectivity. Reminder that we have no selectivity for slow skeletal myosin, because it's the same myosin on there. But we had a 5x selectivity to fast skeletal myosin and nearly a complete selectivity to, which was more important, to smooth myosin. That's probably because of just 2 amino acids in the binding site. This His 561 and this asparagine 654 that cause this really tridentate hydrogen bond to this locked-in water molecule. Now, in fast myosin, one of those is switched out, right? You switch out this histidine for a tyrosine that knocks out one of them. You see a 5x. Where when you switch out the asparagine for both of these, asparagine to actually one amino acid extension of that, knock out both of those hydrogen bonds in smooth muscle, you actually completely remove the binding. So the X-ray helped us to really understand why the selectivity we had and how it probably boils down to just a couple of amino acids in the binding site. So when looking at the farm, does our differences, does that have a difference in pharmacology? And the end of the day is, well, yes, it may not be because of the differences, but we were able to find Aficamten did have a difference in pharmacology. And when one looked at rat, and this is now looking at fractional shortening versus concentration of compounds in a variety of experiments, you can see now the decrease in contractility over concentration for Mavacamten compared to Aficamten. You can see a shallower concentration effect, exactly what we had desired. And then looking at kind of an IC10 to IC50 ratio of 10 versus about 3. So about threefold increase in that ratio. We showed that this ratio kind of looked like it recapitulated over species, and that you can see that this this shallow slope, when you look at free concentration to effect, was the same in rats versus dogs. And later we've been able to show this is very similar in rats, in rat models of HCM, and it's recapitulated across all the species we've looked at in terms of concentration effect. So we're able to really went in with 3 objectives, predicted half-life within 2 weeks. In data I didn't go into, the human projections that we had were 2.8 days for Afi. It was about 3 days at the end of the day, where MAV is 9 days. So we were able to get down to basically a T-half in our range, enough to get to steady state within 2 weeks. We saw much shallower concentration effects, so differentiated pharmacology, and also differentiated in the preclinical pharmacology and the CYP profile in terms of no induction and all of this in this. So, and no notable off targets and then safety preclinically. This also, we were able to show preclinically and work that was a collaborative work done with UC Davis and University of Arizona in Maine Coon cats that have an HCM mutation in myosin binding protein, A31P mutation. So not even in myosin itself. We can see that these cats have a normal range of their fractional shortening is around 50%. So you can see these are hypercontractile rats or cats to begin with. That as we normalize this contractility and get into normal range for these cats, we can completely relieve the gradient of these cats that have obstructive HCM. Exactly what we're going to be measuring in phase 2 in the clinic. So we were, with all this data, we were able to go into the phase 1 study, and our preclinical data translated into healthy participants in that we were able to get to steady state within 2 weeks as projected preclinically. Our concentration, our free concentration effect relationship looked identical in preclinical species. Now looking at ejection fraction in humans versus dogs, you can see that they are nearly identical to one another. So translated across all our species into humans. And then going into really our phase 2 studies where our top line data showed a remarkable response to this, that we were able to, most patients, all but one patient of 14, were able to get complete response in looking at now the decrease in the gradient of obstructive HCM, and then a complete reversal of that upon, cessation of dosing of that. So we saw clinically meaningful reductions in the outflow gradient, no treatment interruptions, no SAEs, no treatment-related SAEs, I should say, reversibility of the drug, and statistically significant improvements of all the other parameters we were looking for. We also had our final dosing between 5 to 20 milligrams in there and our dose titration strategy set up to move to phase 3 and Sequoia HCM. And I could say probably one of the happiest days of my drug discovery life was when I was shown the output of this phase 3 trial for this, and saw that we met our primary endpoint, which our primary endpoint is basically peak VO2, and this is oxygen capacity. And this is kind of a quantitative measure of how well the patients can move and can feel. And we knocked this out of the park in improving the peak VO2 of this, and definitely clinically meaningful differences of that. But not only that, but seeing the p-values for all 10 secondary endpoints, including, how the patients believe they're feeling, the, New York class, the Valsalva, all the gradient impressions, durability, everything. And it's probably, in my career, the best. I give a shout out to my clinicians in such a well-designed and executed study, because I can say it's the most positive study I've seen read out in my years of drug discovery on that. So pretty exciting day in there. So hopefully this will be very beneficial to patients at the end of the day. Safety data also, there were no serious drug-associated events with this, with the major, AEs of being the following, many of which are unrelated to the drug. Quite interesting when you look at hypertension, as I've talked to the clinicians and experts in areas, they think hypertension may be an underdiagnosed problem with some of these patients may have, and that by alleviating these symptoms and making these people healthier, you're actually being able to see the underlying hypertension that may be in these patients and why we may be seeing more hypertension here on there. So that's kind of very interesting that it may be actually showing some underlying causes that could be aggravating in these patients as well. So we're not done when we're still moving on with this. So obviously we have as Dennis mentioned, we have the PDUFA date upcoming. We're in the midst of all of that, but we're also got a lot of really exciting phase 3 trials in progress. Maple HCM, which we're pitting Aficamten against the standard of care, which is a beta blocker, metoprolol. Metoprolol, another of my favorite drugs. I actually take that daily on this, but there's never been a clinical trial, even though it's a standard of care, there's never been a clinical trial of HCM and metoprolol. So we're pitting head to head against the standard of care in this. So in the first trial ever done. So hats off to my clinicians for that. Also looking at a pivotal trial for NHCM. Those that may be aware that Mavacamten just recently announced that they failed that clinical trial. We feel we're very optimistic that having a drug that we believe has a better therapeutic index, we'll be able to get more patients on the right concentrations that will have more success. And I tip my hats to actually my clinic clinicians to another, I think, exquisite design for that trial as well. So we're very hopeful for that trial that we can be hopeful not just for O but for NHCM, and also patients in pediatric with OHCM, and of course our open label for all these patients that once they come off study, we feel it's our duty to keep them on something that we think is should be an agent that will help them out on that as well. So if there's nothing else you take away from this, these are things I want you to really take away from this over 2 decades long search to improve patients' lives. That one, heart disease is really a growing epidemic, right? It's the leading cause of death for people in most racial and ethnic groups in the United States. 1 person dies every 36 seconds from cardiovascular disease. So there's a big need for new therapies here still. And there's substantial medical need here. And it's increased that it hits people of all ages. HCM could hit of all ages. We believe, and I think for much of this, that contractile dysfunction can underlie, for some of these patients, the actual root cause of that disease. And by modulating that, you can could be a viable approach for selective patients, for subsets of patients for these diseases. And most importantly, that, hypothesis-directed research with your eye out for the serendipitous findings at times. And, I get the pleasure of presenting this to you, but it's taking literally hundreds of people and teamwork across these people for any of these successes that we've had. And I want to thank them for that, and really thank all these people, the diverse people that we've had at Cytokinetics, all our collaborators. Amgen was a big collaborator for Omecamtiv development, as an example, scientific advisors, academic collaboration, and most importantly, our patients that inspire us every day to continue to do this work. And I've got to tell you the success stories. Maybe someday I'll come back and share with you the failures at some point from other things. But these are really our successes that we've been able to have over the past couple of decades from our platform. And thanks for your time.
Awesome. Thank you so much, Brett. That was such an amazing presentation. I mean, great to see the phase 3 data from Afi, like really, and patients benefiting from this already. That's basically the basically summit of what you can achieve as a medicinal chemist, right? So
absolutely. Yeah. That's why it's time. As I've mentioned to this, it's time for me to retire. I'll soon be retiring at Cytokinetics.
We're sad to see you go, actually, Brett. Yeah, we do have a lot of audience questions here, which I want to get through a few. I think the most upvoted question here was, what are your thoughts on tissue exposure in the context of medicinal chemistry design? Specifically, how can we improve targeting specific tissues such as the heart through drug design strategies?
Yeah, I think that's a big question, and I think that's the next frontier of drug hunting. I think that's first going to be more important for large molecules, and we're seeing some of that come into the clinic now. I'll go to maybe I'll push a company, Avidity, that's doing phasoscapulohumeral muscular dystrophy, where they're looking at RNA technologies being targeted to through kind of the iron receptors and things. So they're using antibodies targeting, kind of RNAs. So I think you're first going to see tissues selectivity, I think, through larger molecules that may have a longer effect, if you can. So you can get to things that don't need to be there every day, right? So you can get things delivered specifically to a tissue that can do their effects over long periods of time, and you just need to replenish, occasionally. There may be some, like some degraders or some kind of muscle things that can do that as well. That you may need to dose infrequently and go through intramuscular to do all of that. So I think that's kind of the first frontier, and we have to see, how those end up working and learn from that. I think it may be a little more difficult. I think, from getting an oral drug, there's still a lot we need to learn from basic fundamentals to how to deliver that and what are the targets. I think that's around the corner. Don't get me wrong. I think I'll hopefully see that in my lifetime. But I think we're going to have to learn what are we targeting. There's going to have to be specific receptors we're targeting that are selective to the tissues that we want. How can we put drugs that target those specific selectors? There are other people that may think differently. I'll go to Paul Wender's work that he does at Stanford, that he has I would say more empiric ways of targeting tissues on that. But I think there's still a lot of science we have to understand, whether it's through the basic science or the empiricism. We have to learn a little bit more to what are the specific toolboxes we have that target what we need. And we're a long ways from those toolboxes.
Yeah. No. Yeah. Thank you for those insights. Yeah, that's definitely a fascinating question. I've been thinking a lot about brain targeting, of course, during my career. That's another interesting tissue to target. Another question here from Marco. You talked about the Omecamtiv discovery story. Was that actually structurally enabled? You later showed the X-ray structure Was the
Yeah.
campaign structurally enabled, or did you do
Not at
like?
all. Not at all. The we didn't know what the heck we were doing. In terms of what we knew is we were binding to what's called the S1 fragment of myosin through basically you can do limited proteolysis of myosin and you can get to different fragments and you can do even ITC. You can do experiments that show that you're getting affinity and where you're binding to for that, as well as having functional activity. So we knew we were binding to the S1 fragment of myosin. And that's basically all we knew in terms of Omecamtiv. And in terms of the optimization, it was done through these more functional ATPase type of assays to optimize and then not have any of the off-target that we were wanting there. So it was a more classical approach, I would say, to drug discovery. And the X-ray crystal structure came years later after that was actually a molecule into the clinic on that. But it does help us think about it. And I will say that everything we've learned in all of our sarcomeric programs throughout the years that we've done have built on one another to help us, in our designs and how we think about drug discovery.
Yeah, that makes total sense. Maybe two more questions. I know we're already kind of over time, but if you have some time, Brad,
Sure.
how critical is the selectivity between cardiac and skeletal myosin, given that in clinical settings, the inhibition of contractility is monitored and controlled no more than IC20?
I think it's insignificant, to be honest with you. The cardiac seems to be so much more sensitive to these modulators than the skeletal muscle. And I have a hypothesis for this that I have no data to support. But I'll tell you my hypothesis anyway. I think the heart being under kind of, you don't control your heart rate, right? Where you've got complete control of most of your skeletal muscle function through the CNS. And I think because of that and the CNS feedback you get from your skeletal muscle, that it's not really as sensitive as the cardiac is to these modulators. So you need a little bit more. So I think the amount of either activation or inhibition you get on the cardiac that's just kind of tweaking the system is irrelevant to the skeletal system, and that your CNS feedback on that is kind of like, oh, you're maybe getting a couple molecules less of, more or less contractility per calcium. We can make up for that, and there's absolutely nothing to skeletal. So I think it's pretty insignificant in terms of your way to one-to-one.
Awesome. Yeah, thank you. And maybe last question. I mean, we have a lot of open questions, but I think we have to wrap up. So Srinivasan is saying, great talk. Curious to know which method did the team use for human dose projections? Was it allometric scaling or IVE with PK?
Yes. Answer to that is yes. We used all methods.
Oh, all methods. Okay.
Yeah, and the actual kind of 3 days was really amalgam of all the methods that were used.
Yeah, so I mean, it was actually a pretty accurate prediction, which is great.
Spot on. Yeah.
Spot on.
We're not always that spot on, but for this case, we happen to be.
Yeah. Another great thing about Afi, right? It translated well.
Everything translated, so that's why. Hopefully, you see the reason it's my favorite molecule.
Absolutely, absolutely. Yeah. So with that, thank you a lot, Brent, for this super educational seminar. Really, I learned a lot. Everybody here enjoyed it. So yeah, thanks also for sticking around to answer all the audience questions. Really appreciate your insights. And yeah.
Definitely my pleasure to be here. I want to thank everybody who came on. I was worried nobody would, so I'm really happy that you listened to my story.
No, I mean, it was super exciting. And yeah, we're going to be back next week with Wendy Young here, talking about the evolution of women's healthcare, a very exciting topic. So I encourage everybody to dial in there as well. And with that, we're dialing off for today. Everybody have a wonderful rest of your week. And thank you again, Brad. Bye-bye.